Today, understanding complex living systems is hard, but does it have to be? I'm Marcus, I'm a biologist.
I'm Phil, I'm a statistician. Together, we discuss the best experiments to cut through biological complexity. Join us to explore the shape of the next experiment. So, hello again, Marcus. Hey, so this time we were talking to Adam Winnifrith, who is co-founder of Evolvia Biosciences, and we were talking to him about some of the work that he did whilst he was a master's student at Oxford University. So what were the key things for you, Marcus?
Well, talking to Adam's just always a pleasure. He's full of enthusiasm and optimism, and excited about all of the different opportunities that new powerful ways of doing experiments can afford. So in general, it's just a really, really fun discussion, and talking about all sorts of things to do with how we need to be using AI in biology, but then at the same time how important statistics are. He really went into detail as well about the actual biological system that he's working on. So I thought that was really fascinating to dive into. Yeah, definitely. Yeah, for me, it's really fun talking to Adam. I think we've both said before, he's kind of sickeningly clever guy, just a very motivated early career scientist. So it's just his enthusiasm is incredible. And I love the focus on antibiotic resistance, because the story of the development of penicillin, which also happened at Oxford University, it's always kind of fascinated me, and I've always wondered how much quicker we could have got there with using design of experiments. Yeah. So I hope everybody enjoys this episode with Adam Winnifrith. So welcome, Adam Winnifrith, to the next experiment. I know I've known you for a few years, and I think Marcus has known you for even longer. For the benefit of our listeners and viewers, do you want to give a little background to yourself, a little introduction?
Well, thank you so much for having me, Phil and Marcus. I am incredibly excited to come and chat. And, I'm very excited to have a lot of good banter and laughs. I don't quite know how we collectively manage to make just talking about data and science so much fun. Sure. So a bit of background about me. I have I've absolutely always been fascinated by many, many areas of science, and have always liked thinking about how we can use science to solve really big problems in the world. And in particular, got really interested in the intersection between biology, mathematics, and machine learning, and how we can integrate all of those to build better world models of the world. And over the past 6 or 7 years, that whole field has absolutely exploded on so many different angles.
Sure.
And I've dived into loads of projects involved in that. So I did some work on using generative AI for protein design. I obviously done some great work with you guys on trying to collect data really fast. And I'm currently exploring the kind of world models that some of the frontier AI models are learning about the world, and actually how they're learning, and are they learning based on the data, and what can we learn from them even. And in the not too dear future, a lot of these models will understand biology, they'll understand maybe even humans better than us, and can we learn something from them? And that's been a bit of my journey. Just very practically, I studied biochemistry at undergraduate masters. I, alongside that, have built a startup that has been through Y Combinator, and we are increasingly interested in what AI models are learning about the world.
Wow, there's a lot to dig into there. I'm really looking forward to it.
Fantastic, Adam. Thanks very much for your intro to yourself. I think, obviously, on this podcast, on the next experiment, we're talking a lot about experimentation and how we can use experimentation to best effect. We've done some work together, as you've said, where we've been looking at how we can use high dimensional
experimentation, multi-parametric experimentation, to try and explore an assay system to best effect. But when did you first hear about those kind of methods in particular, about like exploring high dimensional space using experiments?
To the best of my memory, I first heard about them when I was reading about drug discovery, and I came across synthase. I genuinely have no idea how, but I remember distinctly watching the synthases. What I imagine they're still up, design of experiments masterclasses, and making notes on them and exploring them through that. And that was the first way I stumbled across them.
Oh, that's actually very cool to hear. I didn't know that. I didn't know that's where you'd started. I'm glad that it's at least sparked a few people's journeys into this sort of space.
It's great to hear, but it's kind of curious that you heard it from an industrial or commercial company rather than through your academic studies from one of your professors, one of your lecturers.
Yeah, indeed. I mean, I think to some extent that is partly my particular undergraduate and master's was in biochemistry, which has a little bit more of experiments in biology and biochemistry. Typically, we've decided that they're incredibly difficult to do. And so we say, well, we can only run so many experiments. And so we'll only try and change one factor at a time. So partly there's sort of a historical hardware experimental issue that we maybe talk about why that barrier might be breaking down in terms of implementing it. And I imagine partly there's also maybe a tendency to not like maths and to not like statistics and to be a little bit scared of it. I think there's one of my favorite greatest lessons at school was being able to have a maths teacher who loved statistics and got the intuition behind it and got the sort of let me explain it in a really, really helpful and in a way that shows why it's really important.
Huh.
Great.
That's really cool, because I mean, often I think there are some real hang-ups that you kind of have to try and get over when starting to work in a much more sort of statistically structured kind of way. But it sounds like actually you didn't maybe personally have those hang-ups yourself. You were quite keen to get into this kind of way of thinking.
Yes, absolutely. I loved. Yeah, we know there's a huge opportunity to understand the world better when you combine maths and machine learning and apply that to really, really tough problems in all the hard sciences. And you say, how can I more rigorously understand the system and the assumptions that I'm making? Because in any experiment that you do, you're making big assumptions about the world. And stats is mainly the hardest stats is actually understanding assumptions. So what assumptions am I making about the system that I'm exploring? And from that flows a little bit of maths that you then apply, but it's mainly about the assumptions. And also, to be honest, really, really honestly, partly as well, it's because the stats questions in A-level maths were Mark's gold mines. Basically, because you could get these huge at maths A-level in the UK, you could say, which is sort of the exam you would take when you're 18, you would have these really complicated differentiation questions, and which were maybe 7 or 8 marks, and big differential equations. And you also had hypothesis testing questions that were also 7 marks. But you got a mark in these questions for writing out the null hypothesis. And you also got a mark for saying, we do not accept the null hypothesis as well. And my stats teacher was particularly pedantic about saying, well, we're not going to, we don't reject it or we don't accept things. We do not reject the null hypothesis as opposed to we accept the alternative hypothesis. But you got loads of marks for really, really easy things. And so I was like, these are great. Why we love stats. Stats gives me easy marks in A-level maths. I get a mark for just understanding about what the null and alternative hypothesis is. So maybe there was a little bit of that as well.
Adam, yeah. if I didn't know you better, and actually that your tongue very firmly in cheek when saying these things, whilst I'm sure that some of it is true at the same time, then I'd be a little bit dismayed right now. But I think I know exactly where you're coming from.
Yeah, and I'm loving hearing that you enjoy the pedantry of statisticians. That's unusual.
No, it's important. It's important.
I mean, maybe it's reflecting a certain rigor of thought, though, isn't it? And I think, I guess at the heart of what it's all about is trying to sift what we can actually have some genuine, rigorous confidence about from all of the stuff which would otherwise mislead us and our imperfect human reasoning, right? And I think that's probably what's at the heart of it. So I kind of laugh a little bit at the pedantry that can sometimes creep in, but I think it comes from a very sound place. It's like, look, and unless we're doing this kind of thing, then we can't really know stuff. And so it's a worthy pursuit, isn't it, to try and get to a point where we can know stuff.
Yes. Yeah. I mean, we very quickly got to the philosophy of science and like, what does it mean to know things and what does it mean to know which might be a little bit beyond the scope of what we want to. Actually, I mean, it's something at the heart of what we want to talk about today. Like, what does it mean to know something, and how can we be confident about it? But all of science is building models about the world and then updating those models as you receive new information. As is the human brain. Just it's we make predictions about the world, and then when they're wrong, we update our world models.
Well, some of us do. I mean, I don't think that's a universal trait, but maybe now we won't stray from experimental design into the philosophy of science and then all the way through to politics. But I think maybe we'll just leave that there.
Let's leave that.
But I mean, in that. When we're seeking knowledge, do you think biology represents a particularly special case? Do you think there's certain features of, the system that has resulted from billions of years of evolution? Do you think that then presents, particularly unique challenges that we need to overcome?
Yeah, especially versus chemistry. So you did biochemistry, so you can you've got a perspective unique here that you can really see. How does it differ from some of these other sciences?
Now, I don't want to make any particular form of science sound harder or different from the others. I think they're all equally challenging and complex in many ways. But I think the clearest analogy that I think most people would agree on is that a lot of the way that we do research on living systems or biology, however you decide to do that, whether that's on an ecosystem level or whether it's down to sort of the biochemical level, and to some extent those lines are becoming, to give people context, those lines are being somewhat blurred because a brief history of sciences was biochemistry as a science used to be very, very, let me analyze reactions of particular proteins in a test tube, which is a little bit about what we did. But whereas biology was, let me take a living organism and break it down. But increasingly, the fields of molecular biology have emerged, genetics, and that has meant they've all sort of mushed together a little bit. But still, even though they're mushed together, I think largely most people would say that the study of biology, biochemistry is about trying to maybe sort of look at a computer and you take a part out and you go, well, what changed? And did it what stopped working? And therefore we can infer what the thing that we took out does, by seeing what broke. And it's sort of we're taking a very complicated system. We don't really understand how it works, and we're sort of trying to break different pieces, or maybe we add two of these things. We've worked out how to make this thing, and so now I'm going to give my computer an extra bit of RAM. What happens to the computer? How does it change? Let's give it two of the things. In comparison to engineering, where we have been building things from the bottom up, we understand the system, we've built them, we've designed them, and so again, those lines are being somewhat blurred. There's increasingly people who are trying to engineer and build things from the ground up in biology. I mean, Craig Venter's synthetic cell was still now quite a while ago. But that high-level principle of biology is this crazy complex thing that we're sort of breaking to try and understand, whereas engineering is this. Engineering and physics and chemistry are often these things we can build from the ground up.
Build a bit more systematically.
Yeah.
I've sometimes heard it be something where biology is more like reverse engineering, right?
Yes.
It's like we have an extant thing, and then we try to pick it apart to try and work out how it works, as opposed to we're going to take component parts and create a new thing out of those parts.
Absolutely. But I mean, this is where one of the incredibly exciting things that you guys are talking about, and we're getting to engage in more and more as a scientific community, is the blurring of those lines, and how much we're increasingly being able to build things from the ground up and then engineer incredibly complex things into biology.
Right.
Yeah. So just taking a step back from this kind of philosophical realm, back to the kind of practical. What was the first DOE? What was the first designed experiment that you ever did, Adam?
The first designed experiment that I ever did. So this was on the system that we talked about in the collaboration that we at the University of Oxford in the Lanyon Hog Lab did with you guys at Synthase and Jub. So a brief primer on that bit of work and the background of the why behind why we were doing it, and then I'll tell you more about the particular experiment that I ran. And so the why was that, as I'm sure many people know, drug-resistant bacteria are undermining modern medicine. So antibiotics are these life-saving medicines that we, as humanity, literally stumbled across only about 80 years ago. And they were transformational. They meant that like a common cut or an infection wasn't life-threatening anymore. And so this was a radical change to the human life experience, to the extent that some estimates put the life extension of medicines that treat bacterial infections at about 10 years.
Wow.
But the big problem is that bacteria were like, well, we don't really like dying, and they played a little Uno reverse card on us. And they've worked out a series of mechanisms, and there's many of them, to evade killing by these antibiotics. And it's that evolution and that problem has got to the point where in 2019, the sort of best estimates put the number of deaths directly because an infection was drug resistant at 1.3 million people worldwide. Which, to put that in context for people, because context is always important, that's more than malaria and AIDS combined in that year.
Whoa.
And there's this huge humanitarian and clinical problem. And one of the things that we really need to get better at is designing and bringing to patients new medicines to treat bacterial infections a lot faster.
Right.
And because of a set of odd economic incentives that are definitely beyond the scope of what we're chatting about today, the financial incentives for drug makers to make a medicine like an antibiotic are just a less than for maybe developing a cancer drug. So a lot of the innovation for these new medicines are being done in non-profit organizations, whether that's academia or whether that's like focused research organizations. And these guys have a lot less money than Big Pharma, and being able to run and design the whole drug discovery process at a much lower cost, much faster, is an incredibly important goal. And we also need smarter strategies. The bacteria, we've got the low-hanging fruit, and so we need to take smarter strategies, and that means our experiments are more complex. So combined with those two motivations of we need a much faster way to do our drug discovery process, and we need to tackle really complex biology because we've picked out the low-hanging fruit, me and the rest of the Lanyon Hog Lab turned to design of experiments. And we said, we need to use it. Otherwise, we're not gonna move fast enough. We're not gonna move in time.
Right.
And so, with that context set, let me tell you about that first experiment. So, innovative strategy. Point number 2. Bacteria, when they get attacked by antibiotics, freak out. They have what is aptly named a SOS response. So they say, "Flip! I'm under attack." And part of this response is to then develop lots of mutations. Like, well, if I start mutating, then maybe I'll be able to escape this attack on me. And this drives resistance. And there's this very complex enzyme that essentially helps to repair damaged DNA. It unwinds DNA, chops it up, and it's called, in E. coli, which is the bacteria that we wanted to target, it's called REC-BCD. REC-BCD. And so this is the enzyme or the protein target that we wanted to develop a drug to modulate. And the reason this is quite innovative and cool is because if we target it, well, we wouldn't actually be killing the bacteria, we'd be stopping the bacteria evolving resistance.
Right.
And that is a novel approach to the, at the moment there's no drug on the market which explicitly, like, its sole purpose is to stop the emergence of resistance. But it's a really complex enzyme because it unwinds DNA, it chops it up, and no one had developed as you talk about assays, I'm sure your listeners will be familiar with the term assay. No one developed an assay, which is just an experiment that says, in drug discovery, like, does this drug work?
Yeah.
And no one developed an assay that would be suitable for screening this protein for drugs. And that's what we set about doing.
Wow. And so how did design of experiments come into this? You said that there's a big motivation to rethink how you do drug discovery. It needs to be done cheaper, faster. I'm assuming that's where design of experiments comes into it.
Yeah, exactly right. We needed to, one of the big things the gold standard that often is talked about in the drug discovery industry is the notion of a assay, a statistical measure called the Z prime statistic. And what this just means is that the signal to noise ratio in your assay is good enough. It's very wide, and you're very confident that if you notice your enzyme observing a reduction in its activity, you can be very confident that this is because of the drug that you've added to your test tube. And so the big aim of our project was to develop an assay which had a Z prime above 0.5, because that's what's needed to progress it. And when I sat down with Tom, who is the lab lead at Oxford, and Markus at the start of my master's year, I was like, well, we need an assay by the end of the year. We need it now because, like, patients need it, and we need to move this forward. And Tom very kindly smiled at me. He was like, Adam, I appreciate the enthusiasm, but I just wanted you to, like, set the expectation that like, drug discovery and assay development takes experienced pharma teams, like, at least usually 3 or 4 months, and where there's, like, teams of 4 or 5 people, and, like, they've got PhDs already. And so I said, well, that's not good enough. Like, we need this now. And so when Markus and Phil got involved, we said, well, what if we tried this? What if we tried design of experiments? What if we let Adam loose on some robots and some software to see what we could try and do within the scope of a master's project? And that was the sort of motivation behind it. I can talk more about the nitty-gritty details of this.
I mean, so you've got this very complex enzyme. You this complex bit of cellular machinery, we could say, and then somehow you have to get a readout from as to whether that bit of cellular machinery is being broken or not, or being hit by a molecule, right? I mean, that's essentially what we're doing in the assay. So what's the kinds of things that you were looking to optimize in there, and what are the kind of barriers that you had to overcome, and what was the experience of actually running that experiment, and I guess what were the outcomes? Like, what was that whole journey like for you?
Yeah. Firstly, I should say all of this was enabled by you guys being so on board with it, including Tom. So it was only possible because they were so enthusiastic about it. And so that experiment itself, what we said was, well, if a drug's coming along, we need to be able to observe a reduction in the enzyme's activity. the main thing this enzyme does is it goes along DNA, unwinds it, and chops it up. And so we needed a measure of how much DNA is left in the test tube, or double-stranded DNA. And so we found that there was a dye that could bind to double-stranded DNA. And when the enzyme comes along, it chops up the double-stranded DNA, and it means that our fluorescence signal goes down.
Right.
If we inhibit the enzyme, and we can do this by removing some of the cofactors or bits and pieces that are essential for it to function, like electricity for a computer. If we remove the electricity that's a very apt analogy. If we remove the electricity for the enzyme, and I'll explain what that is in a moment. If we remove the electricity for the enzyme, then it stops working, and we can see that this fluorescent signal doesn't go down.
Right.
And the reason it was so complicated is because this enzyme, in order to unwind the DNA, it requires a molecule called ATP. For the biologists, we'll know exactly what that is. It's the energy currency of the cell. Cells use it to many reactions inside the cell use it to power some sort of energy transfer. So it uses ATP to unwind the DNA, and then it also uses a magnesium. Magnesium is very important for the function of this enzyme because that's important in some of the biochemistry of exactly how it moves electrons around to chop up the DNA.
Right.
And so we've already got two things that it needs, and it also needs the DNA as well.
Yeah.
And so very quickly within our little test tube where we were assessing how well this enzyme worked, we had like 5 reagents that were absolutely necessary. We had the enzyme, we had the DNA, we had the DNA dye, we had the ATP, and we had the magnesium.
Right.
And I needed to work out, well, how much enzyme do I add? I needed to work out how much DNA do I add? What type of DNA do I add? This is that's quite important. We need to work out how much dye to add and what's the ratio of DNA to dye as well. And we need to work out how much ATP and how much magnesium to add. And for any chemists listening, they'll know that ATP, its structure, means that it's very amenable to bind into magnesium.
Right.
Because there's a bunch of phosphate groups which have negatively charged oxygens on, which are very happy hanging out with the positively charged magnesium.
Right.
Which means that now we had a system where the amount of ATP and magnesium had what design of experiments people would like to call a two-factor interaction. The amount of ATP there impacts how the magnesium affects the thing that we really care about, which is the enzyme unwinding DNA.
Right.
Yeah.
Right. So you have these two different factors, and we know from theory they should be important, right? We know enough about this enzyme that both magnesium and ATP are going to be important things. But then actually, we don't know exactly how altering the concentrations of those two things individually or in concert is then going to actually affect the system. And that's the thing that we need to investigate alongside all of these other factors, right?
Yeah, absolutely. And so very early in my project, I was and also the context for listeners as well, the baseline for assessing how this enzyme worked was to create your reaction mix, then you'd take the DNA out and you would run the DNA on sort of what is basically a jelly, and how long the DNA is tells you how it affects how much it runs down the jelly, slightly indirectly.
Yeah.
For all intensive purposes, the main thing is that the length of DNA affects how much it runs down the jelly. And so you could test maybe, because of the size of the jelly, is about 8 different conditions at once. And I found this beautiful paper that with a lot of effort, managed to test holding the ATP constant and changing the magnesium one factor at a time, and then keeping it and holding the magnesium constant and changing the ATP between different levels, and showing this two-factor interaction. And so one of the first experiments I did was to do a designed experiment where I was hand pipetting into little test tubes, and we had about 20 different runs, but I changed the ratio of ATP, magnesium, DNA, and the enzyme all at once in order to try and understand how they might interact.
Right.
Yeah.
And was this like your first foray then into, like, that initial experiment which you were doing by hand with a pipette?
Yeah.
That was your first foray into then trying to do a structured experiment which allows you to modify all of these things simultaneously. And did that,
That experiment was one of my first proper sort
right?
of
And
deep
did that give you some
experiments.
insight, even with the how many runs did you say you could do?
I mean, like 20. I was
Right.
very limited by, and 20 is a strong word because hand pipetting, these reactions weren't necessarily started at the same time. I was doing a lot of serial dilutions, and in order to make all of this fall into place, it was, I mean, to the best of my ability, these were 20 different conditions.
Right.
Mm-hmm.
Right. But then we're then getting to the kind of practicalities of what it actually takes to run these kind of experiments, right? And the fact that you're limited to 20 conditions, and you've now alluded to the fact that there's all sorts of things that probably might have biased those results a little bit, kind of, is speaking to the difficulty of actually getting these things run.
Exactly, 100%. And we did this, and we said, okay, I think we understand the system enough. The assay itself seems to be working in the way that we want it, but now we really want to move fast to see if we can, by the end of this master's project, can we get a assay with a Z prime where we can robustly show that the Z prime is above 0.5. So we could hand it off to somebody to work on it as a drug discovery tool. And we said, well, in order to do that, we're probably going to need to test a lot more conditions. And 2, we were aware that in the hand pipetting, I was using sort of a pre-made buffer from one of the people who provided the enzyme. And
right.
this was slightly problematic because I didn't know whether that was, I couldn't control the concentrations of things in that. I don't know whether that's going to be useful. I don't know whether that's interacting with my dye as well.
Right.
So that was really important to say, if we want to get to this assay that is going to be useful for discovering a drug, we need to scale up the number of different conditions that we're testing, and we also need to be able to control a lot more variables.
Right. So you're basically deconstructing that buffer so that then you actually get control over its different component parts, and you can change them if you need to.
Exactly. And so the thing that we moved on to then is we said, okay, well, we then went and did a big review of the literature, and we found a bunch of papers from 1977 and things like this. And we observed that there was these incredibly hardworking people who managed to find out that potentially a protein called bovine serum albumin might be important. It might stabilize the protein. And so we thought, well, that could be useful for us.
Right.
And we knew that maybe the pH of the enzyme was going to be important, like the pH at which we ran the condition might be important. And we thought that we had some sodium or potassium chloride inside our system. We didn't know which one, and we didn't know how much of it either to use. We also knew that our buffer contained a chemical called DTT, which is a reducing agent. It seems to stabilize in some cases things, but it's also a little bit nasty. It's a little bit hard to handle. Like, if we didn't have to use it, we don't want to use it.
Right.
And we came up with this list of eventually about 11 or 12 different variables that could affect our system, that could affect our assay. And we said, gosh, where on earth do we start with all of this? And this is where we came up with the idea of, well, what if we could, this is where sort of designer experiments really came into its own, especially in combination with having an automation and a piece of hardware that was amenable to supporting that. And for us, that was the Dragonfly from SPT Labtech, and then putting together the two products that you guys help with, Synthase and Jump, and putting that all together. We said, this we're going to need these tools if we're going to be able to explore this very large 12-dimensional space.
Right. And I mean, from my memory, I remember kind of a very enthusiastic call from you and kind of like sort of saying, "Oh yeah, no, that sounds quite interesting." And then just relentless follow up, it's kind of like, a load of energy, a load of enthusiasm, a load of drive to make this thing happen. And maybe this is what Tom Lanyon Hogg was responding to in the first place when you were telling them as a master's student, "Oh no, we're going to get this assay done." And I think, that kind of drive to take on new methods that might help and really try and drive stuff forward and just try stuff out, right? I think that's a real hallmark of this project. But I mean, and I see it elsewhere as well with people who've adopted these kind of methods and really driven them forward. And it's something we've observed before. It's often the younger scientists, it's people coming in looking, with fewer preconceptions about the way that things need to be done. And yeah, actually, often there's some kind of time imperative or some really difficult thing in front of people that then they're going to try and overcome. So we've got these 11 or 12 factors, nice and easy, no problem. What did the experiment actually look like in the end, and what was it like to run it?
So we said the kind of approach that we took for the people who are familiar with drug discovery is somewhat similar to a screening approach in that and then for people familiar to design of experiments, we would call it sort of a trying to explore as much of the spaces as possible.
Right.
Yeah.
And so we, using our intuition from the literature, set some ranges for the different variables that we wanted to explore, and then said, we worked out, the important thing that we did was we worked out that I was able to run maybe two plates in a day, which would give us a total of 720 different individual wells. We wanted to have things in, and then for each condition, we wanted 3 positives and 3 negatives. So with enzyme / without enzyme / with ATP / without ATP. So our sort of positive controls, if I remember correctly from the paper, were one we added. Enzyme was very, very expensive for us, and it was also very low volume. So one we added enzyme and one we didn't have enzyme.
Right.
We'd done a control separately to show that presence of enzyme doesn't affect the DNA fluorescence for the scientists in the audience.
Right.
So we wanted, the main point with there was for each condition we wanted to have 6 different replicates.
Right.
Which meant that we calculated that we could have about 120 different conditions. And then this is where the wonderful stats of designer experiments came in, because we said, okay, well, we can run 120 different conditions. We have 12 different variables. How can we maximally explore this 12-dimensional in space while maximizing our information, the information content that we receive? And very fortunately for us, the state of stats and designer experiments had got to the point where it could cope with that, and it could just say, sure, here is a set of different variables, here is a set of combination of the different variables which will maximally explore that design space.
Right. And that was work that you were doing, in conjunction with Jump and Phil, and working out the right kind of experiment to run, and then you have obviously the joy of running it, which is then trying to take that statistical ideal and interface it with the practical realities of the lab, and actually get those data points which fit within that beautiful multi-dimensional matrix which Phil and Jump have helped you to design, right?
Exactly. And so I feel a little bit I feel very lucky and maybe not super qualified to speak on this, because I was very fortunate in that I had never tried to run a kind of experiment like this without the help of Jump or Synthase.
Right.
So for me, my experience to run this experiment was we designed it, created some of the designs in Jump, created some of the designs for the experiments in Synthase, evaluated the statistical power. A really important process of this is going like, do I have enough statistical power to make the conclusions that I want to be able to make, talking about assumptions.
Yeah.
And then having chosen that, used Synthase to create the robot instructions, in this case for the SPT LabTech or the Dragonfly. And my experience of that was then like I could see the simulation in Synthase of like how the experiment was going to go. And I sort of just logged onto the computer that was running the Dragonfly and like uploaded the instructions that Synthase had generated for me. And like the most maths that I had to do was doing concentration, sort of dilutions of different stocks to make sure that the stock I was adding to the Dragonfly was the one that I wanted. And even then I used Synthase's stock calculator to help me with that. And so the longest time preparing for the experiment was like making sure like the pH for my buffer was right by like setting the pH probe in and out of this buffer.
Yeah. And the thing that's really amazed me about this experiment was the level of sophistication. And I just don't know if you really understand like how sophisticated this is versus what most people are doing with design of experiments, right? You talked about commercial drug discovery. I'm not aware of this kind of sophisticated experimentation being done in commercial drug discovery. So do you think maybe you were just, you were lucky that you didn't know what you didn't know, and just that gave you the bravery to just do something as complex and impressive as this?
That would be, firstly, thank you. I think that is very likely true. I sort of go, well, we need to model these 12 variables. Like we need to understand how the system works. And yes, I had no priors or no previous experience where people had said, no, that's not something that we do, or we only try 4. And being able to model this 12-dimensional space of how the different variables interact was sort of just like we needed to find it. Like we needed to find the global maxima within this very high dimensional space for the assay, so that we could cut its cost and so that we could find it quickly. I think there was no sort of settling for let's just try and pick the local. I mean, yeah, once I knew that the tools were there and that we could do this, I was like, we have to do this. This is just the best way to find the global maxima, and we want the global maxima for our assay.
Right.
Like I'm not accepting anything less.
Right. And so what were the main conclusions that you managed to then draw from this somewhat ambitious and broad-ranging experiment that you ran?
I mean, very helpfully, most of the combinations were rubbish.
Okay.
Very helpfully, so in this very sort of high-dimensional space, most selections of variables gave pretty rubbish Z primes.
Right.
Some of them, it seemed that the DNA, and like the beauty of this was that then I could sort of put a big, one of the beauty things about the sort of jump software as a visualization was being able to put all of these variables very quickly onto a graph and me just look at it.
Yeah.
We're at this
I remember
time where I felt that as a human who understood the system, just being able to look at the graph data, I could very quickly pick up an intuition for what are the important variables and what was going on. And so being able to visualize things very quickly was amazing. So we did that, and we could very quickly see sometimes the DNA was all over the place, and the DNA seemed to have freaked out and wasn't very happy. In some places, the enzyme just like wasn't working, and it didn't seem to like it was very odd. But we found sort of a few like hit conditions. For chemists who are familiar with drug discovery, they'll often talk about chemical space and the idea that there's more sort of more potential chemicals and combination chemicals than there are estimated atoms in the universe.
Right.
And they'll talk about, well, when we do a drug discovery campaign, we'll try and explore chemical space, and we'll start with a screen where we have we cover a wide area of chemical space, and then we nail down on some different areas of chemical space to explore more. In a very similar way, that is what we were doing, but with design space. And I think that's why this set of experiments, as a mental model, it's so easy for drug discovery to adopt, because they are exploring, they appreciate the vastness of chemical space, and they are already trying to explore the vastness of chemical space. And now with these new automation and software tools, we're going to be able to start to explore the vastness of design space in exactly the same way. We're going to be able to screen large areas of very high dimensional design spaces of assays, and we're going to be able to pick like areas of design space that start to look good. And then from there, I played a little bit around with building some models, but like to start with, this like the data was pretty sparse, so our like models didn't have high confidence, and it was easier for us to say, well, this area of design space looks better. And so the sort of follow up to this was then to say, well, we found some hits, some hit conditions, some hit areas of design space, and we want to locally follow them up. And here I sort of let my curiosity run wild a little bit, because I was looking at this and there was a few different conditions that our two top conditions had about 5 different variables that were different between them, but they were giving very different results. And there was some interesting characteristics about one that I really liked and interesting characteristics about the other that I really liked. So one top condition had very low variation / standard deviation in between the different conditions. Every condition was very close together. Whereas one other condition had a really big signal window.
Right.
In one, the noise was really nice, and in one, the signal was really nice.
Right.
So I really wanted one which had both amazing, like really low noise and great signal. And so my follow-up was to design a series of experiments which explored the differences between these two hit conditions and tried to sort of map out the sort of landscape of design space in between them. Like, how do we traverse from one to the other? And is there any path along that landscape where I'm going to get an even better Z prime with high signal and low noise? And when I sort of had done this initial thing, I had these hypotheses that I want to explore. This is when the combination of automation and the software really came into its own, and the tools that are being given to play with. And at this point, I think you guys had you're starting to trust me enough, so you're sort of like, yeah, do experiments. And so I had this free range to go and explore this system, and well, free range in that I was coming into Synthese's offices for about a week or 2 weeks at a time, and so it was very, very short. It was like proper sprints. So for people in industry, I was sort of doing proper experimental sprints of like 1 week or less, maybe even 2 days. Sometimes it was 2 days long. And so I had this week, which was a luxury. A week of time at Synthese was an absolute luxury for me to be able to do experiments. And so I distinctly remember this 1 week where I had designed, statistically designed a bunch of different conditions to explore, programmed them all, simulated them all, and then I'd sort of scheduled them for sort of Monday, Tuesday, Wednesday. And I came in and was able to, in the course of that week, collect, it would have been on the order of sort of 3,000 data points.
Wow.
Exploring the different system very quickly, back to back. And the beauty of it was that in the evenings, like on the train home, I would be able to already have the data and start building models of these using these different data points in Jump very, very, very quickly. And so by Thursday or Friday, I had different hypotheses that I wanted to test and explore and run. And this is actually, it was actually the final experiments that I ran, so the Thursday and Friday experiment, that ended up as like the final figure in the paper, or sort of somewhat final figure in the paper.
Nice.
Where I had said, let's just do this very cool design of experiment where we explored 5 different parameters in a lot more depth. So
right.
More sort of dense sampling of a local area of design space, as we were talking about. And maybe to sort of conclude and to land that sort of period of exploration. 2 points. One, there is, as with any paper, thousands of data points that didn't end up at all in the paper, didn't end up in supplementary, because I was able to run so many experiments. And point number two was that from the sort of screening of design space that we did, we had about 8% of the conditions gave a sort of Z prime above 0.5, so the target Z prime. When we did this more local exploration of design space between those 2 top hits, we managed to, there was 87% of conditions we tested had gone up to a Z prime above zero 0.5. And so we were really understanding this local design space a lot better, and we were able to hone in on to the best of our knowledge, the global optima in this 12-dimensional design space.
Uh-huh. That's great. I love that story. It reminds me of your story, Marcus, of your kind of early experiences of DOE, where you were talking about making like this. The big surprise was how you made concrete progress week by week by week. You know, each time you're learning a little bit more, and you were just gradually building your understanding and your knowledge. Adam's describing day by day by day.
Yeah.
So it's maybe like this is the shift. This is the next generation, right?
Yeah,
I'm not saying you're old, Marcus, but
well, I mean, I think I had a few disadvantages. I think in that I had to do it all by hand, and it was something where I had to grow cells, which always takes a bit longer. But now I've got my excuses out of the way. I think it's quite interesting though, because what I did benefit from, whether it's a benefit or not, is having done science the old-fashioned way first, right? And so I think it hit me particularly hard. Like, oh wow, I can make such fast progress. I think I think with Adam, he like I'll characterize you, Adam, as someone who makes fast progress in life just by sheer persistence and it's like a hell of a lot of energy thrown at things. I was already used to making incredibly slow progress with biology, having done the PhD where nothing worked for a long period of time, like most PhD students, and done OFAT after OFAT and sweeping them in the bin and all that kind of stuff. And what I find fascinating when I hear Adam talk is that actually it's like, well, why wouldn't you do it this way? Of course it's going to work better. Like it's like it's and it's fantastic. It's really, really good. And yeah, I mean, it's exhilarating to hear about like doing experiments during the day and then analyze them on the train on the way home and then do the next ones the next day. Yeah, it is very cool. And I think many thanks as well need to go to other folks. I mean, I had almost nothing to do with this. I should say I just didn't stand in the way. Is my main claim to have helped here. Whereas other folks at Synthase who the ones who are helping Adam to get this work done as well. And help with the software and that kind of thing. And I think many thanks need to go to those guys. Particularly Steve Brown, I think, helped an awful lot with his joint passion for Synthase and Jump. I think that probably helps.
Yeah, absolutely. And I suppose one thing two things to add. One, like this was an entirely sort of project proposed by us in some respect. We came to you guys and were like, can we come and do this with you? Like, would you be game? And both of you were like so up for it. And I think I just to like very clearly speak to the fact that we had a big vision and a big and a challenge to like get in there, and it was something that just worked like really, really, really well. This was never intended as a this was always the heart of it, like a genuine science problem that we wanted solving, and something that like really, really helped. And it was not clear that it was necessarily going to work, and it was
no science ever.
We were like so excited when it just worked so smoothly.
Yeah, I mean, I suspect that there's a fair bit of luck along the way as well. But I think there are a lot of those kind of human factors as well, and serendipity about having the right people come together and that kind of thing. But no, it was very, very cool. So, Adam, I mean, obviously you've had some good experiences with your early interactions with making stuff happen in biology. What do you see, for the future of the field? Like, what's your ideal state of the way that we're going to be doing biology in, I don't know, 5 to 10 years time?
Wow. There is in 5 to 10 years time, gosh, it's going to be, there's in so many ways it's going to be so different, in so many ways it's going to be so much the same. I think that some of the things that are going to be I'll do the relatively similar first. I think like there's still going to be loads of experiments that are really, really finicky to run. It's very important to note that so much of exploratory biology it's very difficult to automate, in that there's like lots of experiments you're doing for the first time. If you're doing truly groundbreaking and innovative experimentation, which so many people are, there's often you don't automate the experiment, you're sort of building it up for yourself. But there's a huge opportunity to create tools and processes that make those new experiments faster.
Right.
So not every experiment will be automated, is sort of the is the one caveat. Biology is complicated, it's finicky. There's not the financial incentive to automate every single experiment.
Right.
Is my caveat to everything else I'm about to go and say.
Right.
But in 5-10 years' time, oh my gosh, there's so many things to be excited about. Firstly, there's this incredible ability to, let's talk about like data first. As we're increasingly realizing that well-curated, high-quality ML data sets and are the thing that's going to better allow us to understand biology. And the tools that we're building to be able to collect better data sets, collect better metadata, are fantastically exciting. So to give, the tools that you guys are building, so Synthase are building to collect and run way more high-dimensional experiments, the tools that automation people are building to be able to run these, and the tools that people like Jump are building to be able to analyze the experiments are absolutely incredible. And then there's people also building very specific, like ML-ready data sets. Some of them are like really well done open source. So the big one is some of the work from the Align Foundation, and they're collecting very clear data sets to solve different problems in biology. And one of their points is that the thing that unlocked AlphaFold, the Nobel Prize winning discovery, or one half of a Nobel Prize winning discovery, was the PDB, and the PDB was a 10 billion 20-year data collection project. And
longer, right? I mean, that dated back to 1973, I think, or something like that. It was a long and very well curated set of data. Yeah,
exactly. And so the question is, what's the next PDB, and what's the next thing that we can do that is specifically collective for machine learning? A very exciting example of that in the UK is OpenBind, and where we're collecting very specific drug ligand structures and affinities. And that data set will be bigger than the PDB itself, and it's going to be openly accessible.
Fantastic.
Which is hugely exciting. So point number one, there's this increasingly the tools and to be able to collect really well curated specific biology data sets. The next thing that's going to be really exciting is, I think in 5 to 10 years' time, not only will we be able to collect data about the biology, but we'll be able to collect metadata about how the experiment was run. And so I think the benefits of every scientist in 10 years' time wearing a body camera that sees what they do in the lab and is fed into a vision language model that then notes it down exactly what you do will be so huge that everyone will just be wearing them. Because then your entire experiment write-up is done for you. It's exactly what you did, and we have all the metadata, and the vision language model can collect all the metadata about which reagent, how you did it, did you make a mistake? And so there's a few people building these systems already. There's a focused research organization which has built a system called Prism, which is similar to this, but I know that a bunch of people are working on that. I think like everyone will be doing that.
Just makes so much sense.
Because I would just you'll collect all the metadata about the experiment, and I'll be able to very clearly track it, and it will be actually what happened. Maybe, and the other thing that's very exciting, maybe we'll have humanoid robots doing some of that, but in 10 years' time, I feel like humanoid robots will be doing a lot of things. Maybe they'll be doing running experiments, and then then the sort of final part of that is, I think, scaled frontier AI models that can analyze all of that data. So models that are taking in all of the metadata, all of the functional data, and maybe we build specific models that model particular biological problems, and then frontier models can make calls, be agentic and use a particular specialized models, whether that's a genome language model, whether that's a model like AlphaFold, or whether that's a model that predicts binding affinity. I think there'll be frontier AI agents that are able to explore all of this data and make hypotheses and work with scientists to run literature reviews. This week, it was last week, Anthropic released their life sciences toolbox, where we can now plug directly into PubMed and Benchling and various other good life science tools. And I just think that combination of very good collection of biological data, very good collection about metadata of what humans can do, and then the tools that can actually analyze all of that data all at once. I'm so excited for the potential for language models to maybe finally create a financial incentive for negative data. Maybe finally the sort of fact that language models can actually read all the negative data and learn from it, there'll be a financial incentive to do so. Those three things combined make the next 10 years incredibly exciting.
Yeah, it's so exciting, and lots to talk about. I think maybe in a future episode of the next experiment, it feels like we could dig into this and talk for hours about all of this stuff. So thanks for leaving us with such an exciting vision of the future, and thanks for joining us, Adam.
No, thank you for having me and for letting us ramble about science.
Cheers.
Cheers.